Hello students, today again we are going to start a new topic related to the human reproductive health chapter. So in this section we are going to talk about infertility, that is the infertility, the incapability to produce the young ones. So let us see what does it mean. See, you can see children that whatever this concept is there, the infertility, in this it is a condition, either it can be in the male or it can be in the female, in which either the male or female they are not able to produce the young ones. If you talk about the male, then the male is not able to produce the proper amount of the motile, the moving sperm cells, as well as if you talk about a female, the female is unable to produce the ovums, the egg cells, or she is able to conceive, or to get pregnant, or to maintain the pregnancy, So let us talk all of these topics. So you can see the potential causes of infertility are, there are a lot of potential causes of infertility are there. So the number one can be the malnutrition, means when the amount of nutrients, the deficiency of nutrients is there in the body. When there is deficiency of nutrients in the body, that is called as malnutrition, and it can be also the cause of infertility. Same way, the genetic abnormality, sometimes some genetic problem can be there, due to that also the infertility may be there. Same way, the abnormal fallopian structures, the fallopian tubes are in an abnormal shape, size, or they are twisted or twirled. So then at that time also infertility problem comes. Sometimes there is some problem of tumor in the pituitary gland in the brain, or tumor in the reproductive organs, then also the gametes are unable to produce, or if the tumor is in pituitary gland, then hormones are not in sufficient quantity so as to activate your reproductive structures. Same way, alcohol or drug abuse, some males they are involved in alcohol or drug abuses, so at that time also the count in the sperm it gets less and the infertility may be there. Same way, aging is a very big major factor nowadays. Due to aging the infertility is there. Then there is one disease, pelvic inflammatory diseases in the females. In the pelvic region, some inflammation or inflammatory problems they lead to the infertility. In the males, the undescended testes, when the testes do not descend into the scrotum, then also this kind of the problem occurs, the infertility problem occurs. Same way, if the scrotum veins, some veins in the scrotum, they are swollen, they have some inflammation. Sometimes in the females, due to dieting habits, due to improper food habits, they don't have so much of body mass is less, fat is less, then also infertility may be there. The developing antibodies against the sperms, sometimes some females they develop antibodies related to their partner's sperm cells. Whatever the immune system of the female is there, it regards the sperms as a pathogen. At that time it generates antibodies, so it is also very harmful and it also leads to infertility. Now some assisted reproductive technologies are there. Now we have talked about infertility. Now how to cure this infertility? Lot of problems are there, so this infertility should be cured, means some solution should be there related to this. So how we will solve these problems? See, these technologies assist infertile individuals. These technologies they are helpful for the infertile individuals, either male or female, who are not able to bear the children, by handling their gametes or embryos outside their bodies in the lab. These technologies they help them to bear the children. Okay, so it is very often that the first type of technology in this section is intrauterine insemination. From the name itself, intrauterine insemination, IUI, you can understand that In this technique, the sperms are taken from a healthy donor or a husband itself. Then a lady is then introduced with those sperm cells. Then these sperm cells taken from the donor or the husband are then introduced in the uterus of a female by the help of catheter. You can see this is a catheter, and by the help of catheter, these sperms, they can be introduced into the uterus. So this can be a very, what you can say, fruitful method. Like this facilitates the fusion of ovum with the sperms. Fusion of ovum with the sperm will be there, and this is helpful for the males which have low sperm count. This is helpful for the males which have low sperm count. In this, female is all right, okay, but the male is infertile. So this is for curing the male infertility. Okay, so this method is helpful for the cure of male infertility. Hope it is clear. Let us talk about the very popular one, that is in vitro fertilization. This in vitro fertilization or test tube baby. In this technique, the fusion of egg is done and the sperm is done. The egg and sperm fusion, they are done in the laboratory, and then they are lead to generate an embryo, and then this embryo is placed into the uterus. You can see the sperm and egg cell are fused, embryo is made into the test tube, then that embryo is implanted into the uterus by the help of some micropipette or catheter. Hope it is clear. It is helpful for the women who have abnormal fallopian tubes. Means here the egg cell is taken from the husband of a lady, and the sperm cell is taken from the husband of a lady, and the egg cell is taken from the lady itself, and then they are fused into a test tube, and then again they are placed into the uterus of the lady. Actually, the problem comes due to the abnormality in the fallopian tube, where the fallopian tubes are not proper and they are not leading the egg cell to move to the uterus. When the fallopian tube in a lady is not working properly, which is blocking the egg cell movement, ovum movement, and that time the egg cell is taken away from the lady through some means. The egg cell is collected, and then that egg cell is fused with the partner's sperm cell into a test tube, and then it is again placed into the female's uterus. So it is a very helpful technique for the females with abnormal fallopian tubes. Next is, children, zygote intrafallopian transfer. What does this technique means? In this technique, 8-celled blastomere stage is taken. An 8-celled blastomere stage is taken. 8-celled blastomere is taken, and then that 8-celled blastomere is inserted into the fallopian tube by the help of a catheter. This is inserted into the fallopian tube, and from the fallopian tube, whatever the blastomere is there, it slides down and then it implants itself into the lining of uterus, that is endometrial lining. So hope it is clear. This technique is also very helpful for the females which are not able to bear the children due to abnormalities in the fallopian tube, or abnormalities in some times to conceive. Now next one is intrauterine transfer. It is the same as the previous one, but here the technique uses 16 cells blastomere. There the 8-celled stage of the blastomere was used. Here the 16-celled blastomere is used. Next one is gamete intrafallopian transfer. In this technique, ovum from a donor woman is taken, and then it is fused with the sperm at a fallopian tube of a desired lady. Suppose there is a lady, she is not able to produce the ovum, then this ovum is taken from a donor woman, and then the sperm from the partner of that particular lady is taken, and these two are fused, and then they are implanted into the desired, that is the subject lady's uterus, where she gets impregnated and then she can bear the child. Actually here the problem was with the lady that she was not able to bear the ovum, she was not able to produce the ovum, so this ovum has been taken from a donor woman. Okay, next is very uh, recent one, children. This nowadays it is being popular, surrogacy. This topic is a very debating issue nowadays. In this method, a woman is making an agreement with a couple. A third lady is there. This third lady is there. She is making an agreement with this couple. And then what is happening, that this couple who was having lot of age or some problem was there with them, these couples, suppose this male's sperm cell and this lady's egg cell or ovum, they are taken, they are fused, they are fertilized, and then the embryo is placed into this third lady, which is a surrogate mother. So this surrogate mother is going to carry this child, carry the pregnancy for this couple, and after some time, after the gestation period, she will give birth to the child for this couple. Hope understood? I think you people have understood. There is one problem, azoospermia. In this, what is happening in this problem? A male who cannot produce normal amount of sperm cell, his sperm count is less. So in this, what he has to do, what he has to undergo, this is an infertility cause into that male. That male is infertile due to this problem. So now these sperm cells which are taken from this male, which are in adequate amount, which are not in normal amount, which are very very less, so during the time of a coitus they cannot make a lady to conceive. So at this time, this male's scrotum is dissected, scrotum is cut, a cut is made in the scrotum of this male, and insertion of medicine is done from some catheter, from some needle into the seminiferous tubule. Whatever the seminiferous tubule, the tubules which produce the sperms, they are now again activated to produce the proper amount of sperm. Whatever the seminiferous tubules were there, which were not able to produce the proper amount of the sperm cells, now they are activated again by the help of some medicines. And these medicines are inserted by making a cut into the scrotum and reaching to the seminiferous tubule. So this technique is very well helpful for the males which are infertile. Hope it is clear, children. Next, let us talk about some techniques which are utilized for detection of abnormalities in a child. A fetus who is still in the mother's womb, his abnormalities and disorders have to be tested so as to make him grow properly. So for that, ultrasonography technique can be utilized, which understands the position and condition of the fetus. Ultrasonography understands the positioning and condition of the fetus. The second one is chromosomal abnormalities. They are tested by amniocentesis. What is amniocentesis? A needle is inserted into the amniotic sac, and then from the amniotic sac the amniotic fluid is taken, and that fluid is tested for the chromosomes. Same way, chorionic villus sampling can be done in this. From the chorion layer, you can see this is the chorion layer. From the chorion layer, whatever the chorion layer is there, from that chorion layer the number of chromosomes can be tested. Okay, some chorion tissues are taken by the help of a needle, and these chorionic tissues, they are tested for chromosomes, whether they are proper in number or not. Same way, whatever the fetus is there which has developed up to so that it has developed its heart, now this fetus heartbeats can be listened by the help of fetoscope, and this fetoscope can measure the heartbeats, which can be 120 to 160 heartbeats per minute. Hope it is clear. I hope you may be motivating the corona warriors, that is your doctors, your cleanliness maintainers, whatever the policeman, and you may be safe at your home. You are continuing your studies, so keep on motivating these people, children. Meanwhile, thank you.